And so the purpose of what I'm going to be talking about now is to kind of give everybody a background of what cancer is, so that we can better understand some of the more interesting talks that'll be coming up after me. So with that in mind, we're going to start with the fundamentals. We're going to start by defining what cancer is. And when we say cancer, it kind of sounds like it's this single entity, when in reality, it's actually just covers a whole bunch of different diseases. And what I mean by that is you got to think that breast cancer is quite a bit different than stomach cancer. And those are different organs under different hormonal regulation and in a different environment. And even within a particular cancer type, we have different cancer subtypes. So breast cancer has different subtypes, different stages of the cancer, different cell types that are involved. And so even within breast cancer, you have a little bit of differences. But even of two different individuals with the same cancer subtypes, they can have a lot of variation as well. And that has to do a little bit with their differences in their genetic makeup. So there's a lot of diversity when we talk about different types of cancer. And for that reason, it's been a very challenging disease to both study and for oncologists to treat. It's also a disease that develops over time. It's not like this just happens overnight in our body. And that presents some challenges as well. What is common across all the different cancer types is that it's composed of cells that have found a way to divide uncontrollably. And what we mean by that is cells normally divide, they normally multiply. But in cancer, they've devised ways to start multiplying really, really, really fast. And that's what starts to produce this tumor, this mass of cells. What's also really important is what distinguishes malignant cancer from a benign tumor. And that's how these cancer cells devise ways to invade tissue and enter our bloodstream and then spread throughout the body. And that's really where some devastating consequences start arising. For us to understand how cancer develops in the first place, what we're going to do is take you back to grade 11 biology and start thinking about DNA, deoxyribonucleic acid. So in each of us, in the 30 trillion cells that we have of many different types, packed inside the nucleus of those cells is DNA. And DNA, what we're going to think about it as, we're going to think about it like it's instructions. So they, just like a blueprint to an engineer, DNA provides the information, the instructions that program cells when they should grow and divide or die at a particularly controlled rate so that our body stays healthy. So DNA are these instructions. What DNA really is what's called nucleotides. So it's these nucleotides that are linked together. And don't get scared by the chemistry. This is the only chemical structure that you'll see today, at least for me. And what's important in DNA is that there's 4 different types of these nucleotides. So adenine, guanine, cytosine, and thymine. For simplicity, we like to just call them by their first letter. So you'll see A, G, C, or T a lot. And each one, they look somewhat similar between the 4 of them, but they have a little bit of a difference in this part of their structure. And that difference in their structure is what gets exploited for the information that's contained within DNA. And so the information that gets transmitted in DNA, we call this the central dogma. And what's important is the order, the sequence of the nucleotides in the DNA. So if you remember, these letters represent the different nucleotides that it could be. So thymine, adenine, guanine, or cytosine. And if we think about a simple gene, so DNA, our DNA, which has millions of base pairs, is broken down into sections called genes. And if we think of the simplest gene possible, let's say it's composed of 6 nucleotides here. Normally it'd be hundreds to thousands of nucleotides. We have this particular order of GCT, ACC. That order is important. That's because DNA gets converted into another molecule called RNA, and the RNA is segmented into different codons. The codons are 3 nucleotides of a precise order. So in this example, we have codon 1 being GCU and codon 2 being ACC. The exact nucleotides that are in the positions here are really important, because if this was CGU instead of GCU, that would be a different codon. The codons are important because they determine the amino acids that get incorporated into proteins. So there's thousands of proteins in our cell, and each one has a specific thing it's supposed to do, a specific job. So some clear examples are immune cells will produce antibodies to help us fight infections. Antibodies are examples of proteins. Or you can think about muscle cells producing protein to give us strength. So proteins are composed of, let's say, 1,000 amino acids, and there's 20 different amino acids that are possible. And the order and the type of amino acid that gets incorporated into a protein is determined by which codons are present in the RNA molecule. So if we look at the simple protein of 2 amino acids, the purple amino acid here is determined by the GCU codon, whereas the orange one is determined by the ACC codon. If this was a different codon, if this was the CGU codon, we might get a blue amino acid. But for this gene, it has the information to tell this protein it should have the purple amino acid in position 1 and the orange one in position 2. Another way we can think about this is it's like the cell has devised its own secret language, in that the nucleotides are like letters, the codons are like words, and the proteins are like sentences. Using that same analogy, this can help us understand what DNA mutations are. So if I'm messaging somebody really fast and I'm not paying attention, I'm gonna make a typo, and that can lead to some confusion or maybe some something hilarious. If we think about it in the same way, if we look at this sentence, use the big bin for the old hat. If we change one letter, so if we change the H to a C, we get an entirely different meaning for that sentence. Use the big bin for the old cat. I'm putting a cat now into the bin. And so there's an entirely different instruction there. In the same way, if we look at a stretch of codons, if we change one nucleotide, so changing the nucleotide in this codon from an A to a T, that's going to produce a different amino acid. And what the end result there is the protein that gets produced from those amino acids is going to look a lot differently. So this is one way that we like to visualize at the atomic level what proteins look like. If we change one of its amino acids, that could mean that the protein, it looks a lot different and it may not function properly. So it either might completely render the protein useless, or the protein might now be doing something different. So it's these mutations that cause changes to the building blocks of the cell, the proteins. That's important because our cell is composed of thousands of proteins, and each one has a specific thing to do. This is a simplified diagram here of what protein networks in our cell look like. And this one in particular are proteins in the green dots that are controlling the cell's proliferative behavior, how fast the cell is dividing. So it has this entire network of proteins that are controlling how fast, what the cell is deciding to do, whether it should grow or whether it should not. So it's got this whole circuitry of signals that are happening between proteins. So the proteins are controlling, and it kind of looks like a roadmap, right? If we mutate one of those proteins, those green dots, if we either take it away or make it do something differently, that totally recircuits everything. And it's going to cause some chaos, a traffic jam. In cancer, mutations happen such that these green dots, this circuitry is changed, and it gives the cell all these signals that it should grow and divide. And so it has this wrong information. We can think about a cell growing and dividing like we're driving a car. So we have control over the car. We can tell the car to go faster if we put on the gas, and we can tell the car to slow down by putting on the brakes. In the same way, a healthy cell has the right balance of knowing when it should grow and when it should not grow. And so this is a normal process. Skin cells go through a continuous cycle of growth and death. In contrast, what happens in cancer is because of the mutations, the cells no longer have that proper control. So they're now getting all these signals that they should be growing and growing and growing, and they're ignoring the signals that are telling them to not grow. So it's a little bit like if someone put a brick on your gas pedal and cut your brake line. So now you have no control over the car, and that leads to a car crash. So in the same way, through mutations, cancer cells acquire the ability to do rapid cell division, so that's the gas pedal always on. They ignore signals that are telling them that they should execute cell death. So they've lost their brake line. And that's, again, like those networks being rewired. That's because the protein networks are being rewired. Some other things that cancer cells will do is that they'll change their surface. So this is because of those mutations, they'll change the surface of how they look like in the body. And this allows them to evade the body's immune system. Normally, our white blood cells will recognize tumors as a foreign entity and will try and destroy it. But by mutating their surface, cancer cells are able to get away from those cops of our body. Additionally, and this is something Dr. Allen will probably touch on, through mutation, cancer cells find a way to recruit blood vessels. This does two important things. It gives the tumor a constant supply of nutrients. So this is important because they're continually growing and dividing. And it also allows them to travel in the bloodstream and metastasize, where the real problems happen. What's also important to realize is that cancer develops over time. It's going to start with one mutation that occurs, maybe by random. And what will continue to happen is over time, it might accumulate more mutations and start to grow faster and faster. And so it evolves over time as it accumulates more mutations, and more cells are also sort of derived because of these mutations. So eventually, it acquires enough mutations that we get a full-blown tumor that's found its way into the bloodstream. So because it develops over time, age is a risk factor for cancer. And that's because as we age, we increase our chances of accumulating mutations. And that's one reason why cancer has become more common in the 20th century, because humans have started to live longer. There are also other risk factors for cancer, of course, environmental and lifestyle factors. Two that we're very familiar with are smoking, of course, and sunlight. So these two things cause mutations to our DNA. This image shows you what ultraviolet light from the sun can do to the thymine nucleotides. It causes a kink that will lead to DNA mutations. Family history is also a component here, because you can inherit mutations that might predispose you to cancer. So BRCA1 and BRCA2 mutations in breast cancer, that's a very familiar one. But what I would say is that family history is a relatively minor risk compared to the others. So we can't do anything about aging. We haven't developed time travel, unfortunately. And we can't choose what family we're born into, so we can't do anything about that. What we can do is try and develop better treatments for cancer. And so that's what all the scientists are doing across the world. And there's this little cycle that happens how we can try and achieve our goals. So it all starts at the bench. This is where I live. So we try and learn at this level what might work to help us treat cancer. If there's a good idea that's generated, then we might try some preclinical testing, see what happens in simple models. If that proves successful, then we might do some translational work and some clinical trials, which will hopefully give us some useful treatments on the bedside. Of course, it doesn't always go according to plan. And so maybe something doesn't work so well at one of these steps. So then we might go back to the bench and learn why that happened and try to fix it, try to get it better. So there's this continuous cycle of trying to improve cancer treatments and design new ideas. And what it really involves is a whole bunch of different scientists from all different disciplines. So you have the biology perspective. We're going to hear more about cancer biology. At the end of the symposium, we're going to see some biophysics work from the graduate students and how that's helping improve cancer detection and treatments. Chemistry, obviously very important for drug design, and math and biostatistics as well as psychology. So how the emotional toll that it takes on a person with cancer and the caregivers, how we can work with that as well, how we can learn from there. And so it's all of these scientists working collaboratively, working together to give us a better understanding of cancer biology, of the diagnosis, of the therapies and preventative measures, so that ultimately we can all get better patient outcomes. So that's your Cancer 101. So that's 15 minutes of what I've had to learn in 10 years as a trainee. So congratulations.